Okay, welcome again. We have completed ELISA. Now tell me if monoclonal antibodies are used. Suppose primary antibodies which reacts with the antigen of the interest, secondary antibody which react with primary antibodies. Okay. So there are two aspects, primary antibodies and secondary antibodies. Clear ହେଲା ପିଲାମାନେ primary antibodies कौन जो मैंने antigen सहित interact करती secondary antibodies कौन जो मैंने primary antibodies सहित interact करती That means ELISA is a fundamental tool of clinical immunology, and the purpose of an ELISA is to determine if a particular protein is present in a sample, such as serum of a person affected by a pathogen, and if so, how much. तामाने बहुत ELISA and PCR भितरे PCR बेसे अर्ली डिटेक्शन कर परिबो केना से जेनेटिक मटेरियल ऊपर कोची केना डीएनए बा आरएनए जतबे प्रोटीन बनौचंती बा प्रोटीन अगेंस्ट जो एंटीबॉडी बनउची बा ए सब जिंस रे तेनो दिटांक इक्वली इंपोर्टेंट बट पीसीआर द्वारा हमें प्रिवेंशन जल्दी कर परिबा एंड एलिसा डिजीज टिके हेई सरला परे जांकी जणा पड़िबो बा अर्ली डिटेक्शन हे परिबो क्लियर हैला प्लान आगु जिबा ELISA is of two types, direct ELISA, which is used to detect antigen, and indirect ELISA, which is done to detect antibody. Lekha, direct ELISA, which is used to detect antigen, indirect ELISA, which is done to detect antibody. Clear है पिलाने रामजी MPC. Have you written these two lines? आगु जाउची It is detected by ELISA. Write down this. Sandwich assay of ELISA, the enzyme is tagged to secondary antibody. Write down sandwich assay and secondary antibody. Okay. Conventional method of disease diagnosis. Option 1, polymerase chain reaction technique. Option 2, recombinant DNA technology. Option 3, serum and urine analysis. Option 4, both 1 and 2. What should be the answer? Go. Serum and urine analysis. Okay, ये conventional था Rasmita. This is conventional, but modern is different. Early detection of disease is not possible with serum or urine analysis. Correct? Then we have to go for higher things. Molecular fingerprints of disease. Molecular fingerprints of disease are detected by molecular diagnostic techniques. Write down this. ELISA is not one. There are so many. But the right answer is molecular diagnostic techniques. Got? Clear? इसको लिख लो. क्वेश्चन लिख लो समझते. Now we'll enter into gene therapy. Suppose ଆମ ପାଖରେ କିଛି defective gene ଅଛି. ତାକୁ ଆମେ cure ହବା ପାଇଁ ଚାହୁଁଛେ. କେମିତି କରିବା? Heading ଦିଅ gene therapy. ଏବେ ଲେଖ gene therapy under ରେ. Gene therapy is a ଆମେ डिफेक्टिव जीन को काटिकी भल जिन्स लगे जहा आमे गोटे भल जिन्स लगे देले भी चलिबो. Gene therapy is the insertion of genes into the individual cells, tissue to treat a disease such as hereditary disease. केना हमर genetic defect अछि जोतरे की गोटे बदमाश म्यूटेंट एलिलीज बहुत प्रोब्लेम करूची. तेणु बा तार काम से करूनी से हमको गोटे फंक्शनल वन द्वारा कनेक्ट करने पड़ो. The biology of human gene therapy is a collection of methods or technique that allows correction of a gene defect that has been diagnosed in a child or embryo. ठीक अछि. तले फर्स्ट लाइन हमें कौन कहते replaced with functional one. And the biology of human gene therapy is a collection of methods or technique that allows correction of a gene defect that has been diagnosed in a child or embryo. In ଯେତେବେଳେ ସେ disease ଟା develop ହେଇ ଯାଇଥାଏ तापरे हमें 100% क्योर आउ पाई ही परियानी. जदि हमें गैमेट बा अर्ली एम्ब्रियो को जदि हमें ठीक करिवा ताहले हम पाखरे सेटा सुविधा हबो. कंप्लीट क्योर हे परियो. नेक्स्ट जनरेशन विल बी फ्री फ्रॉम द डिजीज. किंतु ऑलरेडी जेहेतु डिजीज डेवलप हेई सरलानी तेणु आम नेक्स्ट जेनेरेशन को आमको जीवाक आउ सुविधा नहीं. आमे आउ क्योर हे परिबा नहीं किंतु आमे सप्रेस कर परिबा. केमिति करबा से विषय कथा होचे. A normal gene is inserted into the genome to supplement an abnormal gene. Mutation is not always good. ବୁଝିଲା? Mutation may be harmful. तो गोटे abnormal gene जोटा कि mutation product जेन तुम जानिथिबो sickle cell anemia मध्य ताहले से जो abnormal gene which is disease causing ताको हमें suppress करिवार अछि तेनु गोटे भलो healthy gene हमकु गोटे vector द्वारा introduce करिवार अछि हैला क्लियर हैला से vector से gene टाको नेला एवं टारगेट सेल रे ताको इम्प्लीमेंट कला the generation of a functional protein product can a healthy gene टा कौन करिवो भलो functional protein बनेवो restore the cells to its normal state से डिफेक्ट थिले मध्य सेटाको ठीक ठाक कर दे परिवो क्लियर है जाए सेटा डिफेक्ट थिले मध्य जेहेतु आमे गोटे फॉरेन जिन देले तमे जानी छो हु इज द नॉर्मल वेक्टर or common vector in human is retrovirus or virus. ताले भाइरस द्वारा हमें फॉरेन जीन बा हेल्दी जीन नहीं कि से डिजीज ऑर्गेनिजम्स बा डिजीज लोक भितरे इंट्रोड्यूस करले जाफଳରେ कि से डिजीज टा सप्रेस हो गला एतिक तार कांसेप्ट ऑफ जीन थेरापी ताले हमें एप्लीकेशन ने आ गोटे पार्ट सारले दैट इज जीन थेरापी जदि हमें ब्रॉडली स्पीकिंग कहबा ताले ह्यूमन जीन थेरापी इज ऑफ टू टाइप्स गोटे ला रिप्रोडक्टिव लाइन बा रिप्रोडक्टिव बा जर्म सेल्स रे हमें जीन थेरापी कर परिबा आ गोटे ला सोमाटिक लाइन थेरापी कोटा बेसी इफेक्टिव जर्म लाइन केन एटा नेक्स्ट जनरेशन को आउ हबनी कि सोमाटिक जीन थेरापी फॉर दैट जनरेशन नेक्स्ट जनरेशन ने भी हेई पारे क्लियर हेलो जीन थेरापी केते माने मोर जीन चेंज करले मोर सोमाटिक कोन रिप्रोडक्शन रिप्रोडक्टिव सेल्स रे जीन चेंज होउची कि होउनी किंतु जदि मोर गैमेट्स रे चेंज हेई परिबो ताले नेक्स्ट जनरेशन मोर पिला मानकर आउ डिजीज हबो नाही हला तो दी टाइप रो अप्रोचेस अछि केना प्रथम तो मु बंचिबा दरकार त मोर सोमाटिक लाइन थेरापी करिया पड़िबो इट्स नॉट ए परमानेंट क्योर बट इट इज ए प्रिवेंट इट इज somehow it is a cure ହେଲା 100% क्योर हबनी मतलब बार बार से थेरापी को यूज करिया पड़िबो कि जर्म लाइन थेरापी रे जदि हमें जीवा तले 100% क्योर हो जाई परिबो let us discuss about germline gene therapy in this type of therapy the functional genes or normal genes are introduced into the sperm माने male gamete or ova female gamete and thus integrated into their genome एथिरे जाइगोट फॉर्म हबो एवं जाइगोट जदि भलो हे गला ताले ऑर्गेनिज्म टा कंप्लीटली भलो ही जाई परिबो क्लियर हेलो पिला ने जदि जनकर जाइगोट भलो ताले से ऑर्गेनिज्म इज कंप्लीटली भलो ही जाई परिबो केना हमें समस्ते जाइगोट रु बनीचे हम समस्ते कोड बनीचे जाइगोट रु बनचे कि नहीं तो जदि जाइगोट भलो हे गला ताले जाइगोट रु माइटोटिकली हम समझते बनीचे बा हमर सब ऑर्गन डेवलप हेची ताले विल बी कंप्लीटली क्योर हे परिबा क्योर फ्रॉम द डिजीज हे परिबा बुझी परलो पिला ने विल बी कंप्लीटली क्योर फ्रॉम द डिजीज यू कैन सी मी आल्सो ना देखि परछो तो पिला माने देखि परछो कि नहीं मते ओके आगु जीवी ताले कोटारे चेंज आनले बेटर जीन रे चेंज आनले बेटर ना कोतरे चेंज आनले बेटर पिलाने को चेंज आनले बेटर जीन रे चेंज आनले बेटर सोमाटिक रे ना जर्म सेल्स रे किए कोई परियो सोमाटिक ना जर्म सेल्स कोतरे चेंज आनले बेटर किछि कोना कोटा बेटर सोमाटिक ना जर्म सेल्स कोतरे चेंज आनले बेटर यस वेरी गुड नो पिलाने किछि आंसर देले आ नहीं नहीं सोमाटिक हेले मोर फायदा हबो रश्मि सोमाटिक हेले मोर फायदा हबो देटा मोर सर्वाइवल पई यू आर करेक्ट बट जदि मु जर्म सेल्स रे चेंजेस आनीबी तल सेटा परमानेंट क्योर हबो बुझेला ओके so due to various techniques and ethical reasons the germline therapy remains at the infant stage ବର୍ତ୍ତମାନ germline stage ଏତେ develop ହେଇପାରୁ ନାହିଁ है ना infant stage ରେ ଅଛି ଆଗୁ ଯାଉଛି ତାଲେ जदि आमे एक रे इंट्रोड्यूस कर दे परिबा तले that mouse is completely cured केना एक रु जाइगोट बनेला जाइगोट रु बनिछि हला एबे आस सोमाटिक लाइन थेरापी in this therapy the functional genes are introduced into the cells of the patients the approach is to correct the disease phenotype by treating the somatic cells ता माने कोन बुझि पालो पिलाने जे एटारे मोर बोन मेरो सेल को जदि मु ठीक करिवि ताले मोते से डिजीज ता जो हेतिला जेनेटिकली एटा टिके कम हबो क्लियर हेलो किंतु एथिरे मु 100% क्योर हे परिबि कि नो हे परिबि नाही the changes affect in this gene therapy are non heritable माने नेक्स्ट जनरेशन को सेटा फेरे हबो बुझेला एटी देखो तार ता बॉडी रे जे ट्रांसजीन दिया गला से क्योर ही गला किंतु फ्यूचर रे तार पिला माने से डिजीज रे सफर करिवे केने तार सोमाटिक सेल नेक्स्ट जनरेशन को जाउनी जर्म सेल्स बा रिप्रोडक्टिव सेल्स नेक्स्ट जनरेशन को जाउची जा भी हो पर्टिकुलर पर्सन पई सोमाटिक लाइन थेरापी यूज हबो एंड जदि आमे नेक्स्ट जनरेशन को डिजीज फ्री करिया पई चाहूचे ताले हमको कोटा दरकार हबो ताले हमको जर्म लाइन थेरापी करिवा को पड़िबो जोटा कि परमानेंट क्योर आगु जाउची somatic line therapy can either ex vivo or in vivo ex vivo माने ला cells are modified outside the body and then transplanted back ex vivo माने outside the body in vivo माने genes changes occurs inside the cell जतरे से माने सेल भितरे ही अछंती क्लियर हला इतिटा ओके नाउ we'll enter into next phase of our discussion हला आगु जाउची बुझि पड़ो क्वेश्चन नंबर 1 को the biology of human gene therapy allows the correction of a chromosomal disorder lifestyle disease a genetic defect and infectious disease कोटार करेक्शन हबो क्रोमोसोमल डिसऑर्डर ना जेनेटिक डिफेक्ट कोटा करेक्ट कोटा करेक्ट क्रोमोसोमल डिसऑर्डर ना लाइफस्टाइल डिजीज yes Rasmita you're absolutely correct it's a genetic defect स्पर्म बा ओभारे जदि फॉरेन जीन को इंट्रोड्यूस कल ताहले ताको आमे कोन कहबा सोमाटिक लाइन जीन थेरापी कहबा ना जर्म लाइन जीन थेरापी कहबा ना बोथ कहबा ना नॉन कहबा स्पर्म बा ओभा आर सोमाटिक ना जर्म लाइन any of you Ramji right somatic line gene therapy yes Rasmita it is germ line kineta reproductive okay changes or modification becomes heritable in dash कोटा next generation को जीवो somatic line gene therapy और germ line gene therapy hmm yes germ line gene therapy very good Rasmita कहना reproductive cell ହିଁ ତ next generation को ଯିବ and that is a permanent cure ଯଦି ଆମେ first clinical gene therapy ହେଇଥିଲା 1990 ମସିହାରେ ଗୋଟେ ଛୋଟ ଝିଅ 4 ବର୍ଷ ତାକୁ ଗୋଟେ ଡିଜିଜ ହେଲା ସେ ଡିଜିଜ ର ନାଁ ହେଲା ସିଭିୟର କମ୍ବାଇନ୍ଡ ଇମ୍ୟୁନୋଡେଫିସିଏନ୍ସି ସେ ଡିଜିଜ ର ନାଁ କଣ ପିଲାମାନେ କୁହ ସିଭିୟର କମ୍ବାଇନ୍ଡ ଇମ୍ୟୁନୋଡେଫିସିଏନ୍ସି କ୍ଲିୟର ହେଲା ଏବଂ ଏଇ एसीआईडी डिजीज कौन पाई हुए जानिଛ एटा हुए आडेनोसाइन डियामिनेज ବୋଲି ଗୋଟେ ଜିନ୍ ଅଛି ସେଇଥିର ଡେଫିସିଏନ୍ସି ଯୋଗୁ ସେଟା ହୁଏ କ୍ଲିୟର ହେଲା एडीए ର ଫୁଲ ଫର୍ମ ଲେଖ ଆଡେନୋସାଇନ୍ ଡିଆମିନେଜ ତାର डिफेक्ट ଯୋଗୁ ତାର ଡେଫିସିଏନ୍ସି ଯୋଗୁ এই डिजीज ଟା ହୁଏ ହେଲା କ୍ଲିୟର ହେଲା ଏ ଦିଟା ଲାଇନ ଲେଖି ଦିଅ ଆଗକୁ ଯିବା ଆଗକୁ ଯିବା କୋଉ ଜିନ୍ ର ଡେଫିସିଏନ୍ସି ରେ ଏଟା ହୁଏ ପିଲାମାନେ କୁହ କୋଉ ଜିନ୍ ର ଡେଫିସିଏନ୍ସି ରେ ହୁଏ କୁହ एडीए ଜିନ୍ ହେଲା ଆଗକୁ ଯିବା in some children ପିଲା ତ ଅଲରେଡି ବଡ ହେଇସାରିଲାଣି ଆଉ ତ ଗ୍ୟାମେଟ ଲେବୁଲରେ ନାହିଁ ତାହେଲେ ତାର ଡିଜିଜ କୁ ଆମେ କେମିତି କ୍ୟୁର କରିପାରିବା ଆଉ ତ 100% କରିପାରିବାନି କିନ୍ତୁ ବୋନ ମ୍ୟାରୋ ଟ୍ରାନ୍ସପ୍ଲାଣ୍ଟେସନ we can use enzyme replacement therapy can be used କିନ୍ତୁ ଯଦି ତାର ଆମେ lymphocytes ରେ ଯଦି ଆମେ କଲଚର କରିବା ଆଉଟସାଇଡ ଦ ବଡି ଏବଂ ତାକୁ ଯଦି ଆମେ ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରିବା ତାହେଲେ ବେଟର ହବ କି ନାହିଁ ତେଣୁ ଆମକୁ ସେଗୁଡା ଆପ୍ଲାଏ କରିବାକୁ ପଡିବ ତାହେଲେ ଆମେ କଣ କରିବା ନା ଗୋଟେ ଫଙ୍କସନାଲ ଏଡିଏ ଜିନ୍ ନବା obviously that is cDNA କମ୍ପ୍ଲିମେଣ୍ଟାରୀ DNA ଏବଂ ଗୋଟେ ରିଟ୍ରୋଭାଇରସ ଭେକ୍ଟର ଦ୍ୱାରା ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରିବା lymphocytes ରେ which are cultured and subsequently returned to the patients. This is very very important. Outside the body ଆମେ introduce କରିବା ଏବଂ ତାପରେ reintroduce କରିବା। କିନ୍ତୁ ଏଥିରେ permanent cure ହବନି, periodic infusion କରିବାକୁ ପଡିବ। However, as these cells are not immortal, the patients requires periodic infusion of such genetically engineered lymphocytes, and permanent cure is not possible. Clear ହେଲା ପିଲାମାନେ? Permanent cure not possible. Clear ହେଲା କି ନାହିଁ? ଆଉ ଥରେ କହୁଛି ରଶ୍ମିତା, ଆମେ permanent cure କରିପାରିବା କି permanent cure କରିପାରିବା ନାହିଁ, କାରଣ ସେ କିନ୍ତୁ ଆମର ଆଉ ଅପସନ ନାହିଁ, ପିଲାଟା ତ ବଡ ହେଇଯାଇଛି ଆଉ ତାର ଗ୍ୟାମେଟ କି ଜାଇଗୋଟ ତ ନାହିଁ ଆମ ପାଖରେ ନା କି ଆମେ permanent cure କରିପାରିବା। ତ permanent cure ଆମେ କରିପାରିବା ନାହିଁ। କ୍ଲିୟର ହେଲା ଏଇ ଯାଏ? ତାହେଲେ permanent cure କରିପାରିବାନି କିନ୍ତୁ ବଞ୍ଚେଇ ରଖିବା କଣ କରିବା ନା ତାର lymphocytes ଗୁଡା ଆଣିବା ଏବଂ ସେଇ lymphocytes ରେ ଆମେ foreign gene, healthy ADA gene introduce କରିବା ହେଲା, adenosine deaminase by the help of retrovirus। retrovirus ଗୋଟେ ଭାଇରସ ସହିତ ଭେକ୍ଟର ସେ ନେଇକି ଯିବ ଏବଂ ସେ ଭାଇରସ ଗୋଟେ ଡିଜିଜ କଜିଂ ନ ସେମାନେ ଯେତେବେଳେ ADA gene introduce ହେଇଯିବ ତା ବଡିରେ ତାହେଲେ ସେ ବର୍ତ୍ତମାନ SCID ରେ ଆଉ ସଫର କରୁନି, କିନ୍ତୁ କିଛି ଦିନ ପରେ ସେଟା ସରିଯିବ, ସେ ସେଲ ଗୁଡା ତ ଇମୋର୍ଟାଲ ନୁହଁ ନା, ଫେର ଆଉଥରେ ଏମିତି ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରିବାକୁ ପଡିବ, କାହିଁକି ନା କିଛି ସମୟ ପରେ ସେଟା ସରିଯିବ, ପରମାନେଣ୍ଟ ହେଇପାରିବ ନାହିଁ। କିନ୍ତୁ ଯଦି ଆମେ ରଶ୍ମିତା ଗ୍ୟାମେଟ ବା ଜାଇଗୋଟରେ ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରିଥାନ୍ତେ ବା अर्ली एम्ब्रियोनिक सेल्स ରେ ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରିଥାନ୍ତେ ତାହେଲେ ନେକ୍ସଟ ଜେନେରେସନ ଯଦି କରିବା ତାହେଲେ ନେକ୍ସଟ ଜେନେରେସନ 100% କ୍ୟୁର ହେଇଯିବ ନା, କିନ୍ତୁ ଏଇ ଜେନେରେସନରେ ତ କିଛିଟା କ୍ୟୁର ଦରକାର ନା, ତେଣୁ ପରମାନେଣ୍ଟ କ୍ୟୁର ନଟ୍ ପସିବୁଲ, କିନ୍ତୁ ନେକ୍ସଟ ଜେନେରେସନରେ ଆମେ କରିପାରିବା अर्ली एम्ब्रियोनिक ଷ୍ଟେଜରେ ବା ଗ୍ୟାମେଟରେ ବା ଜାଇଗୋଟରେ। କ୍ଲିୟର ହେଲା ରଶ୍ମିତା ଏଇ ଯାଏ? ଯେତେବେଳେ ସେ ଅର୍ଗାନିଜିମଟା ବଡ ହେଇଯାଇଛି ସେତେବେଳେ ଆଉ କଣ ସେ ଗ୍ୟାମେଟ ଲେବୁଲରେ ଅଛି କି? ଆଉ ଗ୍ୟାମେଟ ଲେବୁଲରେ ନାହିଁ। ତେଣୁ ଗ୍ୟାମେଟ ବା ଜାଇଗୋଟ ବା अर्ली ଷ୍ଟେମ ସେଲରେ ଅଛି କି? ନାହିଁ। ତେଣୁ ଆମେ ତାର ବୋନ ମାରୋ ସେଲ୍ସ ବା ଲିମ୍ଫୋସାଇଟ୍ସ ରେ କିଛିଟା ଯୋଉମାନେ ସେଲ୍ସ ବନାନ୍ତି ସେଇମାନଙ୍କ ପାଖରେ ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରିବା ଯାହାଫଳରେ କି ଲିମିଟେଡ କ୍ୟୁର ପସିବୁଲ। Tell disease and gene therapy. Scientists are focusing on disease caused by single gene defect, such as cystic fibrosis, hemophilia, muscular dystrophy, sickle cell anemia. ଏଗୁଡ଼ାରେ କ'ଣ ହେଇଛି ନା ଯଦି ଆମେ ଗୋଟେ ହେଲ୍ଦି ଜିନ୍ ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରିଦେବା ତାହେଲେ କ୍ୟୁର ହେଇଯାଇ ପାରିବ। କ୍ୟାନ୍ସର ଉପରେ ବି ଆମେ ଲଟ୍ ଅଫ୍ ରିସର୍ଚ୍ଚ କରୁଛେ ହେଲା, କ୍ଲିୟର ହେଲା ପିଲାମାନେ? କ୍ୟାନ୍ସର ଉପରେ ବି ଆମେ ଲଟ୍ ଅଫ୍ ରିସର୍ଚ୍ଚ କରୁଛେ ଯାହାଫଳରେ କି ପର୍ସନ ଜଣକ 100% क्योर ହେଇପାରିବ। जेके विके कॉलेज यू कंटिन्यू, ओके, बीच बीच में आप चले जाते हो, आगे चले। जीन थेरापी ओवर, समझते बुझीला रश्मिता बुझीला बोधहुए बेले बेले पिलांगर क्लास फ्लास रही जाउची तेणु पढ़ाउछंती फिर आसुछंती, किंतु you communicate all your friends and we want to see in more number ହେଲା। ଆମେ ଏତେ ପରିଶ୍ରମ କରୁଛେ ତ ମାନେ ଏତେ ପରିଶ୍ରମ କରି ମୁଁ ପିପିଟି ବନେଇଛି ହେଲା, ଆଗକୁ ଯିବା। ଏଗୁଡା ସୁନର ଲେଟର ତମର ଦରକାର ହବ। କେତେବେଳେ ଦରକାର ହବ? ଯେତେବେଳେ ତମର କ୍ଲାସ ସରିଯିବ ସେତେବେଳେ ଅଛି ନା ବାୟୋଟେକ୍ନୋଲୋଜିରେ। ତ in spite of so many efforts, the biology of GT remains complex. Many techniques need improvement. Gene therapy. You can communicate who is sharing the এটা তুমি ভিডিও ଦେଖିପାରିବ ହେଲା ରଶ୍ମି, there is one video group ଆମର DHS ର ଭିଡିଓଜ ରେ ବି ଅପଲୋଡ ହଉଛି ହେଲା, ତ ୟୁ କ୍ୟାନ୍ ସି ଡିରେକ୍ଟଲି ଦେयर ଏଇ ଯୋଉ କ୍ଲାସଟା ରେକର୍ଡ ହଉଛି and it is uploaded, so you can get the better picture from that. ତମେ ବାର ବାର ଏଇଟା ଦେଖିପାରିବ ଏଇ ଭିଡିଓ ବି। The genetic link of many disease is to be understood more fully before gene therapy can be used appropriately। ତାମାନେ ଆମକୁ ଆହୁରି ରିଡ୍ କରିବାର ଅଛି। ତ ଟ୍ରାନ୍ସଫର ଅଫ ଏଇଠି ଗୋଟେ କ୍ୱେଶ୍ଚିନ୍ ଆସେ, ହଁ ମୁଁ ଲିଙ୍କ ଦେଇଦେବି DHSC ର ଲିଙ୍କ ଦେଇଦେବି by the end of the class। ତ ଏଇଟା ସମସ୍ତେ ଲେଖ। ହଁ no problem, you can see the videos also। ଜେନେଟିକାଲି ମୋଡିଫାଏଡ DNA ଯଦି ଗୋଟେ ଗୋଟେ ଭଲ ଜିନିଷଟେ ତମେ କୋଉଠି ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କଲ ୟୁକାରିଓଟିକ ସେଲ କୁ, ତାହେଲେ এই ପ୍ରୋସେସ କୁ ଆମେ କଣ କହିବା? ଟ୍ରାନ୍ସଫେକ୍ସନ। ଲେଖ, the process is called transfection। It is into the eukaryotic cell, remember this। ଯେତେବେଳେ ତମେ ଜିନ୍ ଗନ୍ ଦ୍ୱାରା ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରିପାରିବ, ମାନେ ବମ୍ବାର୍ଡମେଣ୍ଟ କରିପାରିବ, ଆ ଭିତରେ ଗୋଟେ ଜିନ୍ ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରି ଇଲେକ୍ଟ୍ରୋପୋରେସନ ପୋର ଫର୍ମ କରିକି ଜିନ୍ କୁ ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରିପାରିବ, ମାନେ ଗୋଟେ ହେଲ୍ଦି ଜିନ୍ କୁ କେମିତି ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରାଯାଏ ସେଟା ଆମେ କହିଛେ। ଏବଂ ଭାଇରାଲ ଭେକ୍ଟର ମଧ୍ୟ ୟୁଜ କରାଯାଏ ବୋଲି ମୁଁ କହିଥିଲି। ମୋଷ୍ଟ କମନଲି ଆମେ କହିଥିଲେ ଭାଇରାଲ ଭେକ୍ଟର ଇଜ ଅଲସୋ ୟୁଜ୍ଡ ହେଲା, ଭାଇରସ ଦ୍ୱାରା ଆମେ ୟୁଜ କରିପାରିବା। ତିନିଟା ମେଥଡ କହିଲି, ଭାଇରସ ଦ୍ୱାରା ବି ଆମେ ୟୁଜ କରିପାରିବା। କ୍ଲିୟର ହେଲା? ଭାଇରାଲ ମେଥଡ ମଧ୍ୟ ଆମେ ୟୁଜ କରିପାରିବା। କ୍ଲିୟର ହେଲା କି ନାହିଁ ପିଲାମାନେ? ଲେଖ, ଭାଇରାଲ ମେଥଡ ମଧ୍ୟ ଆମେ ୟୁଜ କରିପାରିବା। ଆଗକୁ ଯାଉଛି, ସପୋଜ ଏଟା ଗୋଟେ ପ୍ଲାଜମା ମେମ୍ବ୍ରେନ୍, ଆ ଭିତରେ ଆମେ ଗୋଟେ ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ କରିବା, ତାହେଲେ ଏଇ ଜିନ୍ ଚାରିପଟେ ଲିପିଡ ଅଛି, ଲିପିଡ ଆଣ୍ଡ ପ୍ଲାଜମା ମେମ୍ବ୍ରେନ୍ ଭାଇ ଭାଇ, ତେଣୁ ତା ଭିତରେ ଇଣ୍ଟ୍ରୋଡ୍ୟୁସ ହେଇପାରିବ ନା, ଲିପିଡ କୋଟ୍ସ ଦେଲେ ଇଜିଲି ଆମେ କରିପାରିବା। involving the complex polymers ଏବଂ ସାଇଲେନ୍ସିଂ ଅଫ ଜିନ୍ ଦ୍ୱାରା ବି ଆମେ ଡିଜିଜ କୁ କମେଇ ପାରିବା। ତ we have completed disease। ଏବେ ଆସ କୁହ ମତେ, सोमाटिक लाइन जीन थेरापी रे एक्स विवो हुए ना इन विवो हुए ना बोथ? इधर और ना एनीवन? यस, यस Rasmita, now you are very clear, either ex vivo or in vivo। ADA stands for adenosine deamine, adenyl deamine, adenosine deaminase, adenosine deamino acids। Yes, adenosine deaminase, very good। को डिजीज हुए deficiency of ADA causes which disease? Option 1 AIDS, option 2 SCID, option 3 yes severe combined immunodeficiency। Very good Rasmita, all three are correct। खनम समझ में आ रहा है कि नहीं? सस्ता खनम। Now we'll enter into transgenic animals. Transgenic माने कौन? Transgenic माने है genetically engineered. Genetically engineered. Transgene. Can I say genetically modified organisms? Yes. Genetically modified organisms. So animals that have their own genome and had their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Done. Foreign gene. Give some examples. Transgenic rat use हुए, rabbit use हुए, pigs, sheep, cows, fishes. किंतु सबुठु अधिका काहा उपरे काम हुए? Who animal transgenic animal? That is mice. Human being रो most of the research काहा उपरे हुए? ना mice उपरे हुए. Clear हेलो? एडी लेखो एटा गोटे NEET रो question. 95% of all existing transgenic animals are mice. Clear हेलो? आगे चले. ट्रांसजीन जहाँ पाखरे गोटे फॉरेन जीन अच्छी. फॉरेन जीन कहीं इंट्रोड्यूस करिछ? कारण फॉरेन जीन इंट्रोड्यूस करले से ऑर्गेनिज्म रो कैरेक्टरिस्टिक्स बढ़ी जिवो, गुरुत्व बढ़ी जिवो. फॉरेन जीन ने किछि गोटे भलो कैरेक्टर अच्छी जोथ पे ताको हमें इंट्रोड्यूस करिछ. माइस, रैट, रैबिट, पिग्स. पिग माने कोथेरा सोर्स जानछ त पिलाने? पिग माने कहार सोर्स कहले देखिया. पिग माने ला ऑर्गन रो सोर्स. पिग माने ले ऑर्गन रो सोर्स. लिखि दियो पिग माने ले ऑर्गन रो सोर्स. Hello. Now you can see the rat, rabbit, pig, sheep, cows, and fishes. Out of that, which is more important, that is mice. କେତେଗୁଡେ example ଆମେ କହୁଛେ transgenic animals are more sensitive, more developed, and therefore we are using them for our benefit, especially to design, to study how genes are regulated and how they affect ଏବେ ଏଠି ଦେଖ transgenic animals can be specifically designed to study the genes or କାହିଁକିନା ସେମାନେ sensitive ତାର ଗୋଟେ healthy ଲୋକ ଅଛି ଗୋଟେ unhealthy ଲୋକ ଅଛି and medicine ର impact healthy ଉପରେ ଯେମିତି ପଡିବ unhealthy ଉପରେ ସେମିତି ପଡିବନି କାହିଁକି ନା unhealthy ତ ମୂଳରୁ ସେ disease ଲାଗୁଛି କିନ୍ତୁ healthy ସାଙ୍ଗେ ସାଙ୍ଗେ weak ଦେଖାଯିବ ବା ସାଙ୍ଗେ ସାଙ୍ଗେ ତାପରେ effect ହବ and transgenic organisms are well developed sensitive and they are more control environment ରେ ଥାନ୍ତି ତ ତାଙ୍କ ଉପରେ ରିସର୍ଚ୍ଚ କରାଯାଏ ହେଲା ତେଣୁ ଇନସୁଲିନ ହଉ ହରମୋନ ହଉ ଡ୍ରଗ୍ସ ହଉ ତାଙ୍କ ଉପରେ ହିଁ ରିସର୍ଚ୍ଚ କରାଯାଏ କ୍ଲିୟର ହେଲା ପିଲାମାନେ ଏବେ ଆସ ନେକ୍ସଟ ପାର୍ଟକୁ ଯିବା ଆଉ କଣ କରାଯାଏ transgenic animals are designed to increase our understanding of the genes contributed to development of disease and specifically made to serve as models for human diseases so that investigation of new treatments for disease is made possible. ତାହେଲେ ଆମେ disease ର study କରିପାରିବା pathophysiology study କରିପାରିବା ତା ସହିତ ଆଉ କଣ କରିପାରିବା ନା disease କେମିତି develop ହଉଛି ମୋ body ଭିତରେ ସେଟା ଆମେ study କରିପାରିବା by using transgenic animals especially mice ଆଉ କଣ କରିପାରିବା ନା new treatment ମଧ୍ୟ ଆମେ ତା ଉପରେ କରିପାରିବା ଗୋଟେ ମେଡିସିନ ଦେଲେ ତାକୁ କଣ ସାଙ୍ଗେ ସାଙ୍ଗେ ହ୍ୟୁମାନ ବିଙ୍ଗ ଉପରେ ଆପ୍ଲାଏ କରାଯାଏ କି କରାଯାଏନି ତାକୁ ପ୍ରଥମେ ଟ୍ରାନ୍ସଜେନିକ ଅର୍ଗାନିଜମ୍ସ ମାନଙ୍କ ଉପରେ ଆପ୍ଲାଏ କରାଯାଏ ଆଣ୍ଡ ଅଲ୍ଟିମେଟଲି ହ୍ୟୁମାନ ବିଙ୍ଗ ଉପରେ ଆପ୍ଲାଏ କରାଯାଏ ବୁଝିହେଲା Today, transgenic animals exist for many human beings like cancer, cystic fibrosis, ବହୁତ ଜାଗାରେ ଆମେ आउ कौन बेनिफिट हुए पिलाने को बायोलॉजिकल प्रोडक्ट वी कैन गेट धर गोटे गाई रो क्षीरटा ह्यूमन बीइंग रो क्षीर सहित नुह किंतु जदि हम पाखरे एमिति गोटे मिलिबो जोटा कि एग्जैक्टली ह्यूमन बीइंग रो क्षीर सहित समान सेई टाइप रो गाई ताले इट इज बेटर the medicine required to treat certain human disease can contain biological products, but such products are often expensive to produce. Transgenic animal द्वारा हमें biological products we can create by introducing the gene, that particular gene which does code for a particular product such as human protein. ଯେମିତି କି antitrypsin to cure or to treat emphysema. ताମାନେ ଆମେ medicine बनै परिबा ता सहित ଆମେ milk भी बनै परिबा. Similarly, attempts are made to treatment of phenylketonuria and cystic fibrosis. जदि आमे 1997 को जिबा सेई टाइम रे गोटे फर्स्ट ट्रांसजेनिक काउ रोजी बना हेतिला जी कि ह्यूमन प्रोटीन एनरिच्ड मिल्क बनै पड़तला जोते कि 2.4 grams per liter human protein enriched milk thila and this milk contains human alpha lactoalbumin and was nutritionally more balanced product. I mean normal गाई ठारू रोजीर मिल्क human baby ପାଇଁ is more valuable and जोटा नॉर्मल मिल्क मिलुची अनट्रीटेड काउज द्वारा ताठारू एटा बेटर. Alpha lactalbumin is rich in essential amino acid that is the dominant protein in human milk. जदि हमें देखिबा what are the essential amino acids मैं तुमको आगुरी भी कहिथिली 8टा essential amino acid अछि and what are they? Methionine, tryptophan, tyrosine, valine, lysine, phenylalanine, isoleucine, and leucine. These are not produced in our body. We have to take it from other animals or other plants. Jadi gote protein re all these essential amino acids are there, then it is good. And it is one of the dominant protein in human milk, lactalbumin, alpha lactalbumin. Aku chhadi ki transgenic mice are being often used to test the safety of vaccines before they are used on humans. मतलब कहली 95% research transgenic mice द्वारा हुए. पोलियो vaccines मध्य transgenic mice द्वारा हमें टेस्ट करते तार सेफ्टी टेस्ट मध्य कराई. जदि हमें कहबा who is the source of organs, then it is transgenic pig. If successful and found to be reliable, they could replace the use of monkeys to test and safety of batches of vaccines. Kaina ami bhabutile je chimpanzee, gorilla, monkey, they're close to us, tanka ami use kariba, but they may suffer or they may be bigger organisms. We should not use them. We should go to the equal sensitivity found in mice. तो mice गुड़ाक भी आमे commonly use करुचे. Next part को जिबा chemical safety testing. This is known as toxicity or safety testing. कौनसी chemical तार side effect अच्छी जदि side effect अच्छी केते dose रे बा केते concentration ने हम ताको use कर परिबा as medicine that we should see. This is known as toxicity testing, and this procedure is the same as the use for testing toxicity of drugs. ଯେମିତି କି ଗୋଟେ ପାଣିରେ ଭଲ କି ଖରାପ there is a certain level of toxic substances in the water. ତ ତାକୁ ବି ଆମକୁ ଟେଷ୍ଟ କରିବାକୁ ପଡିବ whether that water can be used for drinking purpose or not. Transgenic animals that carry genes which make them more sensitive. ତ ମୁଁ କହିଥିଲି they are well developed, they are sensitive, and they are often used for testing. କାରଣ ସେମାନେ ଯାହାର ସେନ୍ସିଟିଭିଟି ନାହିଁ ସେ ବେଟର ବୁଝିପାରିବନି ସେ ଟକ୍ସିସିଟିଟା ଜାଣିପାରିବ ନାହିଁ ତେଣୁ ଏମାନେ ନନ୍ ଟ୍ରାନ୍ସଜେନିକ୍ ଆନିମଲ୍ସ ଠାରୁ ବେଟର. They are then exposed to toxic substances and the effects are studied. Toxicity testing in such animals will allow us to obtain results in less time. ବର୍ତ୍ତମାନ इंडिया गवर्नमेंट गोटे कमिटी बनैछि एटा बहुत थर आसलानी क्वेश्चन एंड दैट कमिटी इज कॉल्ड इट्स रिलेटेड टू इंडिया नॉट टू द वर्ल्ड that is called genetic engineering approval committee and this committee look into misuse of DNA manipulation and safety of introducing genetically modified organisms for public services. ଯେମିତି କି GESC बीटी कॉटन को अलाउ करिछि बट बीटी ब्रिंजल को अलाउ करने कारण बीटी ब्रिंजल इज एडिबल किंतु बीटी कॉटन हमें खाऊनी तेणु बट इट इज क्रिएटिंग लिटिल प्रॉब्लम फॉर कैटल एंड अदर pet animals but it is not harmful because cotton we are not eating. But BT brinjal is not allowed by this GESC to be used in India. Can you tell me what is the full form of GESC? Genetic Engineering Approval Committee. Now again some of the MCQs. Question number 1, animals that possess express an extra or foreign gene are called transgenic animals, hybrid animals, transitional animals, trisomic animals. Please answer. जिसके पास एक्स्ट्रा और फॉरेन और ट्रांस जीन अच्छी सही टाइप रो एनिमल्स को हमें कौन कहवा? Anybody? Ramji? Quick, give the answer. Baripada, JKBK, Sukruli. The answer is transgenic animals. Question number 2. The first transgenic cow is, its name is what? Roji. Okay. Chemical safety testing is also known as vaccine safety testing, toxicity testing, disease testing, antigen testing. Toxicity testing. Now we are entering into the last part of our discussion. And these are stem cells. What do you mean by stem cells? In botany or in plant, we are saying them as callus, group of undifferentiated dividing cells. And in zoology, we are saying stem cells as undifferentiated cells found in most, if not all, multicellular animals. So this is a word related to animals. Got? Stem cells are characterized by the ability to go through numerous mitotic cycles. କାରଣ ଆମର ସମସ୍ତେ ଜାଇଗୋଟରୁ ଆମେ ବନିଛେ ତେଣୁ ଏଟା ଗୋଟେ ମଲ୍ଟିସେଲୁଲାର ଫର୍ମ ଏବଂ this multicellular structure is produced by repeated mitosis. But these stem cells are in undifferentiated state. जोड़ाक आमे बॉटनी रे आमे कहिथिले callus. The normal cells produced by mitosis will go for differentiation into different types of cell types and ultimately different organs. But stem cells are in undifferentiated state. Stem cells are thus responsible in future for development, for repair of adult tissues and cancer when control over them is lost. Cancer is uncontrolled cell division. Stem cell research has the potential to revolutionize the future of medicine with the ability to regenerate damaged or diseased organs. एमाने सेल्फ रिन्यूअल करंती धर किछि डैमेज हे जानती आउ किछि ताको बनै दियंती एवं सेमाने को पाखरे रिजेनरेशन पावर अछि बा सेलुलर टोटिपोटेंसी अछि टू रिजेनरेट इनटू ए कंप्लीट इंडिविजुअल और टू रिजेनरेट इनटू कंप्लीट organs. We know liver has the power of cellular potency. Okay. I told you cellular potency is of three types. One is totipotency, one is pluripotency, and third one is multipotency. And we'll discuss all these things one after other. In plants, cellular totipotency is there. What does it suggest? It suggests the plant cell regenerates into a new individual. Got? Suppose this is a stem cell which is having the power of self-renewal, means one stem cell can produce another stem cell, but at times it differentiate into mature cell also. That means stem cells are in G1 and matured cells in G0. Differentiated cell is in G0 state. Now you see the next diagram. Potency specifies the differentiation potential of the stem cells. Totipotency. It is a Latin word, totus. total or entire or omnipotent, means these stem cells can construct a complete viable organism. Cells produced by first few divisions of zygote and even those from morula are also totipotent. In plants, somatic cells or most of these cells are totipotent enough to produce the new plant. But in animals, the zygote or even those from morula are totipotent. They can produce or construct a complete viable organism. Got? Now, the next word is pluripotent. Latin word pluros means several or many. Plural word also you know, singular and plural like that. Pluripotent stem cells are the descendants of totipotent cells and can differentiate into nearly all types of cells, but not into complete organism. Cells derived from any of the three germ layers like ectoderm, endoderm, or mesoderm, embryonic stem cells are pluripotent and they are producing the organs. Got? You can see here, they can produce different organs, but not the organism. They can be taken from ectoderm, endoderm, and mesoderm. And we know ectoderm produces certain organs, mesoderm produces certain organs, endoderm produces certain organs, but they can produce the entire organism. Now the last part of our discussion is multipotent. Multipotent stem cells can differentiate into a number of cells, but only those of a closely related family of cells. ଯେମିତିକି mesenchymal stem cells multipotent and they differentiate into bone, cartilage, connective tissue or adipose tissue, means their closure. Or some cells, they produce hemopoietic stem cells which produce all the cells of blood. Got? Now the next part is unipotent. Unipotent cells can produce only one cell type. माने सेमाने निजोकु निजे बनेइबे. But have the property of self renewal. माने गोटे प्रकार matured cells बनेइबे. But बहुत प्रकार matured cells बनेइ परिबे नाही. Which distinguishes them from non-stem cells. In mammals, there are two broad types of stem cells. One is embryonic stem cells, and the second one is adult stem cells. After maturity, certain stem cells are also there. They will go on producing or adding new cells. Embryonic stem cells are isolated from inner cell mass of blastocysts. Embryonic stem cells are pluripotent and can give rise to three primary germ layers, ectoderm, endoderm, and mesoderm. They are pluripotent, not totipotent. And when given the necessary stimulation, they can develop into more than 200 cells types of the adult body. तामाने जदि मोर किडनी दरकार मु सेई टाइप रो सेल्स को आनी मोर किडनी बनै परिबी बा लिवर बनै परिबी जदि मु सेई एम्ब्रियोनिक स्टेम सेल को आज पर्यंत कल्चर करी थांती बा नेक्स्ट जेनरेशन ने तमे जदि कल्चर करिवो ताले यू कैन गेट योर ओन किडनी लिवर और heart even different types of organs even and you can reintroduce like a car when one vehicle is going to the shop ame gote part ku kadhi au gote part lagai dauche in the same way we can use them ethi ame gote jinsa dekheiche je most of the blood cells can be produced from multi-potential hematopoietic stem cells ରୁ ଯୋଉଥିରେ ଆମେ WBC ବି ବନେଇ ପାରିବା ବା RBC ବି ବନେଇ ପାରିବା embryonic stem cells are immortal cells we can cultivate for time so long time in artificial conditions and they are the source of large number of cells means they are the source of large number of organs when they will differentiate. Adult stem cells are found in various tissues of children as well as adults. Any cell which is found in the body of developed animals that has the ability to divide and create another cells like it is called somatic stem cells. Okay? So adult stem cells are mainly responsible for repair and replenishing gut. Most of the adult stem cells are multipotent. The red bone marrow is rich source of adult stem cells. For example, mesenchymal stem cells, adipocyte stem cells, endothelial stem cells, etc. Do you know pluripotency are rare and generally in small number, occur in umbilical cord? Write down this. Pluripotent adult stem cells are rare and generally in small number occur in umbilical cord. Mother रो child सहित जोटा connect करे fetus को. Now we'll start MCQ. It is the last part of our discussion. All of you are ready to solve all these MCQ. Write down. Stem cells are found in all multicellular animals, few multicellular animals, most multicellular animals, or all the three. Can any of you answer this? Stem cells are found in all multicellular animals, few multicellular animals, or most multicellular animals. What should be the answer? Got Baripada, Katak, Moyurbhanj, Ganjam. What should be the answer? Bhubaneswar. The answer is stem cells are present most multicellular animals which have the power of division and regeneration. The second question, stem cells are responsible for development, repair, undifferentiation state, or 1 and 2. They're responsible for repair and development. Already I told you, stem cells taken from umbilical cord can be transformed into specialized cells to replace myocytes or neurons or perhaps the entire organism through cell culture. A number of adult stem cells therapies already exist. We can make organs also. आगे चले एइथिरे स्टेम सेल सब अच्छी pluripotent stem cells are descendant from totipotent cells. Identify the unipotent stem cells from the following: cells of morula, embryonic stem cells, muscle stem cells, or homeopoietic stem cells. The answer is muscle stem cells. It is making only muscle tissue. Therefore, it is unipotent stem cells. Okay. Now tell me, entire biotechnology application will revise now. We know the green revolution in our country is due to the teamwork of William Goud, Norman Borlaug, Khorana, or M.S. Swaminathan. Yes, father of green revolution in India is M.S. Swaminathan. सबको समझ में आया? Next. Revolution, which can be obtained by using genetic engineering method for increasing in food production and reduce of chemical fertilizer. एटा जेनेटिक इंजीनियरिंग द्वारा जो नेक्स्ट रिवोल्यूशन आसीबो ता ना कोन दबा? कैन आई से क्रोमोसोम रिवोल्यूशन? कैन आई से मॉडर्न ग्रीन रिवोल्यूशन? कैन आई से जीन रिवोल्यूशन? कैन आई से एडवांस ग्रीन रिवोल्यूशन? Yes, the answer is gene ra change hochi, transgenics hochi. Therefore, the answer is gene revolution because it is genetic engineering. Very good. आगे चले पिलान टे कथा हबो microbes, plants, animals whose genes are modified by genetic engineering are called as option 1 GEO, GMO, GMM, GEM. The more common word is genetically modified organisms. I'm writing here genetically modified organisms. Very good. Ella GMO mane kono? Genetically modified organisms. Genetically modified organisms or plants are tolerant to abiotic stresses, help to reduce the post-harvest loss, most susceptible to pest disease. No, no, no. But they enhance the nutritional value. Therefore, third option is not correct. They are resistant to diseases rather than susceptible. Clear hello? Genetically modified plants. I mean, total discussion karuche biotechnology application ro. You can write the answer if you can't answer even. Ramji done. MPC done. JKVK सुक्रुली डन. आगे चले. Bt toxin produced by a bacterium called as, yes, Bacillus thuringiensis. Ranji, very good. Insects like Lepidopterans, Coleopterans can killed by toxins released from Bt. And what is the full form? Yes, Ramji, Bacillus thuringiensis. Bt toxin protein remains inactive in Bt because it is not getting alkaline environment in the bacteria. But in beetles, dipterans, and lepidopterans, it will get the alkaline environment in the midgut so that poration occurs and the organism can survive. In the gut of insects, Beta-toxin protein turns into active form by alkaline pH, acidic pH, absence of epithelial cells, more proteases. The answer is alkaline pH. Very good, Ramji. Disease controlled by toxin coded by gene CRY1AC and CRY2AB. Can any of you tell me CRY1AC and CRY2AB is cotton bollworm? If I'll say CRY1AB, then it is corn borer. सबको समझ में आ रहा है? CRY means is the cry gene. आगे चले. अगर हम ऐसे लिखेंगे तो it is cry protein. Cry gene produces crystal protein, which is toxic in alkaline environment. Disease controlled by the toxin coded by the gene CRY1AB is dash. I told you right now. Is it cotton bollworm or corn boll? No. CRY2AB nahi, Ramji. CRY2AB I am not saying. I am saying CRY1AB. Then what's the answer, Ramji? It is corn stem borer. Because cry genes are highly specific. Yes. Activity toxins binds to surface of dash, midgut of epithelial cells, labrum, hypopharynx, mandible. Yes, Ramji, you are absolutely correct. It is 1. JKVK and others should join. आगे चले. Baripada, you can join also. Roots of tobacco plants get infected by a nematode. This is 1. Agrobacterium, Longidorus, Melodogyne incognitia, Gyphenema. What's the answer? Tobacco, RNA interference, gene silencing re ame पढ़िचे. Melodogyne incognitia. Zoology re बोधे पढ़ेइबे and you can understand. Write down. It is Melodogyne incognitia. राम जी समझ में आ गया? आगे चले। सुक्रुली। Okay, very good. The process takes place in all eukaryotic organisms as a method of cellular defense is RNA interference, RNA translation, RNA transcription, RNA replication. What should the answer? RNA interference. Very good. Bismagiri Ganjam, Ramji, higher secondary. You people are very good. The sources of complementary RNA from for the RNA interference is infection by fungi, infection by virus having RNA, transposomes, or both 2 and 3. You are introducing by the help of viruses and transposomes. Okay, jumping genes. Transposomes are called jumping genes. Okay. Even we can introduce by the genetic engineer natural agrobacterium vectors also. आगे चले. This can be called as strands of RNA and antisense of RNA, both club to form double-stranded RNA. Yes, yes. Commercial synthesis of hormone is possible by utilizing cloned DNA, cloned RNA, cloned mRNA 2 and 3. Cloned DNA. Generally, DNA is used because commercial synthesis for human being, insulin production, I told you. Therefore, it is cloned DNA. Total number of recombinant therapeutics have been approved worldwide for human use are, I told you today, it is about 30. Yes, yes, Ramji. Very good. Number of recombinant therapeutics presently marketed in India. Actually, now it is more, but as per NCERT, it is 12 only. So you go by NCERT. Done. 12. Out of 30, 12 in India. Identify the incorrect statement with respect to genetically modified plants. I told you earlier also, it reduces post-harvest losses. That is correct. Crops less tolerant to stress. No. Improved nutritional value of the food. Yes. And increase the crop yields. ठीक है. Therefore, the answer is crops less tolerant to stress, as told by Ramji. Very good. Now, human insulin is now prepared in large scale by using bacteria called DAS, Acetobacter, Bagiota, Salmonella, E. coli. I told you today, the humulin or human insulin is produced by DAS. Can any of you tell me? बोलिए. It is E. coli. Very good. Which company? Eli Lilly. It is American. And they are producing DAS, human insulin,
Human lipase, human amylase, human urate. What should be the answer? Bully, insulin A, insulin B. I told you also today. Yes, the answer is option 2, human insulin or humulin. Can any of you tell me insulin A and B chains extracted from E. coli are combined by creating which bond because of cysteine amino acids? Is it dipeptide or is it disulfide or is it dihydrogen bonds or dinitrogen bonds? I told you this is insulin A, this is insulin B, and something is there like this and like this also. Yes, very good, Ganjam, Ramji Higher Secondary Schools. Bismagiri Ganjam, you are absolutely correct. The answer is two disulfide bonds. Thank you, all of you, listening me so long. So thank you, thank you.